My name is Joe Zackular. I'm an assistant professor in pathology and laboratory medicine at the Children's Hospital of Philadelphia and the University of Pennsylvania. I'm a basic researcher, and our lab focuses on the gut microbiota and enteric pathogens like Clostridioides difficile. We are, working to harness the microbiome as well as develop new therapeutics to handle this really urgent public health threat, which is C. diff.
And we're going to be talking about your study in the mRNA vaccine for mice that ended up being successful. Can you just tell us how you plan on moving towards the next steps from animal models to human models?
When it comes to a really complex infection like C. difficile, I like to start with animal models that recapitulate what we see in humans so that we can study the immune response, the efficacy of, treatments like vaccines. So ultimately thinking about safety, thinking about immunogenicity, thinking about the effectiveness of these strategies for, taking on a challenging infection like C. diff. Animal models are very valuable, but of course, the ultimate goal is to bring therapies like these vaccines to humans. And so a lot of the work that we did in the animal models, we did by design with, physiologically relevant concentrations of these mRNA vaccines that would be safe to use in humans, validated by all of the work that has been done with the COVID-19 vaccines. And what we're working towards now is using other animal models, larger animal models, to look at immunogenicity and safety in these animal models like hamsters and non-human primates. And ultimately, we would hope to bring this to phase 1 clinical trials to begin to look at the efficacy and safety in humans. And, the hope is that the exciting promise that we see in animal models, like the mouse model that we use in the study, applicable to what we'll see in humans, and will provide a robust tool for this really problematic infection.
How do you predict this vaccine's ability to clear existing C. diff bacteria and improve outcomes for patients that have recurrent infection?
C. diff is a really challenging and uniquely challenging pathogen for us to take on. It lives a really complex life cycle, and so C. diff is a vegetative cell, so a bacterium that produces two really potent toxins that cause disease. But C. diff also makes a spore. And this spore is really, really hard to kill, can survive outside of the body, but even when it's inside the body, it can be really challenging for antibiotics to kill it. It can be challenging for our own immune systems to kill it. And so C. diff's ability to kind of oscillate between this vegetative lifestyle when it can be growing and producing toxins and then this dormant spore, make it really challenging. The goal would be to prevent all C. diff from being able to colonize and cause infection. But that challenging, it's an organism that lives in this relatively hard to get to for our immune system part of our body, the lumen of the gut, and it doesn't invade. And so being able to prevent it from colonizing, of course, can be challenging. And so when we're attacking it with this vaccine, what we found is that we aren't able to prevent the initial colonization and outgrowth of this pathogen. However, what we are able to find in this, in these animal models is that we're able to dramatically stunt the pathogenicity and the disease that's caused by this, really problematic infection. And so, in the animal models, we're using a highly, highly virulent strain of this pathogen that causes 100% of the mice to die. But when we provide this vaccine, we're able to prevent these and prevent all mortality, which is really remarkable in terms of its efficacy in the future. How, as we continue to improve upon this platform, so this study is a really nice proof of principle of the power of the mRNA vaccine platform for this infection. But ultimately, in the future, we would hope to be able to either decrease the amount of bacteria that can colonize or, ultimately maybe prevent colonization altogether. It's a lofty goal, but that would be the hope. And these mRNA vaccines, just a little bit more background, take on this really complex enteric pathogen, right? So it's a vegetative cell, it can be a spore. It's within the lumen, but it's producing these toxins that then go on to cause damage and inflammation in our epithelial cells. That's a challenge because there's a lot of aspects to this infection or this pathogen you have to try to overcome when you're thinking about developing a vaccine. But this mRNA vaccine platform allows us to create multivalent vaccines very readily. And so what we were able to do in this paper was make a tetravalent vaccine that targets both of the really important toxins that cause disease with C. diff, but also target the cell itself as well as the spore. And so, this is a really exciting proof of principle that we're able to create a multivalent vaccine that can be highly successful for preventing disease, for an infection like C. diff. But ultimately, in the future, we're hopeful that we can add even more targets against the cell, against the spore, against other virulence factors that might allow us to prevent initial infection or at least, clear even quicker.